थोड़ा light excellent. So thank you all for bringing it here on a Sunday. We are in Prithvi House today because the Prithvi Festival is going on and there's a play in the theatre. But don't worry, we are part of the Prithvi Festival also, and we have arranged a small theatre here as well. So I hope there are at least 20 people here because we need 20 participants. 20 participants?
We need at least 20 participants for this game called protein folding.
So we're going to play a game.
Everybody's going to play.
It's a pleasure to introduce Professor Ravi Venkatramani from the Department of Chemical Sciences in TIFR. Ravi, I'm going to let you introduce your background if you want to. We're keeping this chai-wise very informal, as you know. What we will, let people come. What we certainly will tell you is that in case you're coming for the first time, Chai and Why happens on the first Sunday of every month here, either at Prithvi Theatre, usually sometimes in the cafe outside or sometimes in Prithvi House over here when the theatre is not available. Thankfully, Prithvi gives it to us essentially for free, which is great. It happens on the third Sunday of the month at Ruparel College in Matunga, our mid-month venue. And if a month has a fifth Sunday, we are typically doing a session from a lab in TIFR, which is online only. So far, we used to be at a school, but we still don't have permission to go back to the school. So right now, fifth Sunday sessions, if and when months of fifth Sunday, have been online. As things improve, we will see how that develops. So today we're going to be doing protein folding, but just in case you're curious, the next Chai and Why, which is 2 weeks from now, the third Sunday of November, you haven't found a speaker, right? Which means I will do it myself. I always have some emergency Chai and Why topics. So this one's going to be about the chem history and the chem mystery of ammonia. Okay, ammonia is a sort of smelly gas you've probably heard about, or maybe you smelled in a chemistry lab somewhere. But it's got a beautiful story, and we'll talk about it 2 weeks from now. I'll do a Chai and Why on that. Too much of chemistry, no? Anyway, so doesn't matter. Chemistry is fun. We're all made up of atoms.
Right? This is biology.
This is biology. So it's interesting. You know, all the chemistry Nobel prizes and the very
life science
Nobel prizes, they go to chemists who are working in biology usually. Yeah. So anyway, Ravi, all yours. Introduce yourself. I'm going to sit there and monitor that the videos not collapsing and things like this. We are live on YouTube.
Yeah, sure.
So audience who are watching us online, if you have questions, please put it in the chat, and I'll try and read them out to you, or we'll take them at the end. Obviously, online audience, you don't get chai. Okay, the audience here gets chai, so you will have to make your own chai, I guess. You don't get to play the game as well. So with that, it's all yours.
Yeah. Okay. Thank you, Arnav. So my name is Ravi Venkatramani. So I am in the Department of Chemical Sciences at Tata Institute of Fundamental Research. My own background is a little odd. I started out as a physicist. I got my PhD in physics. But while I was getting my PhD, I noticed that I was working more and more with chemists. Right. Then I ended up figuring out a career where I was working with chemists all my life. But unfortunately, in the chemistry department, I was always working on more biological problems. So at the end of the day, when I was applying for jobs, it was not clear which department would accept me. Ultimately, it was Department of Chemical Sciences. It said, come in, welcome, join, etc. And I'm comfortable there at this point. So today, as Arnav says, chemistry, biology, physics, these are all, these boundaries are disappearing rapidly. So today, I'll be actually telling you something about proteins, right, which are biological molecules inside our body. So this talk is assuming that you have no background other than the fact that you heard about proteins in the general media or whatever it is. And so if some of you already know some of these ideas, that's completely fine. I mean, I'll move towards, hopefully you'll get something more out of it as we move towards the final slides. Yeah, so I'm going to be talking about the protein universe, something about how diverse and how big it is, and how artificial intelligence is really enabling us to make breakthroughs in understanding the protein universe. So let's start with the most basic slide.
And this is also celebrating the breakthrough prize.
Yeah.
Our last one, we did the physics breakthrough prize. We're doing the life science. We did part of the life science breakthrough prize also, and this is again part of the life science.
Indeed. So the 2023 life sciences breakthrough prize was given precisely for this problem actually. So in some sense, it is also a celebration of that. Okay, so you must have heard about proteins. I'm sure mummy is telling you to eat eggs, for example, or drink milk at least, right? I mean, like, and because eggs and milk contain lots of protein. This is our first introduction to protein in our life, right? You must have, other than that, you must have heard about, hopefully some of the younger guys must know, basically, that if you want to build muscles, my muscles are not that big actually, unfortunately, you need more proteins actually. So I should be eating more protein, right? Other than that, in the general media, you'll find lots of products that are advertised which are protein enriched. This is just a example from Amul, right? Protein enriched drink, but then you also have protein enriched shampoos, etc. in the market. So proteins are around us, right? But do we really understand what they are, right? What are they basically? What do they look like? What is their role in our lives, etc.? Why do we actually care about these supplements, drinks, etc.? Okay, so I'm going to start with a very general, broad analogy, actually, right? So I'm going to start talking about something that we all understand. Let's think about a city like Bombay, right? And let's think about how we kind of classify the city and understand the city. So city, is made out of localities. Like I come from Kolaba, right? But there's Bandra, there is Parel, there is regions further down in the suburbs, actually, right? So all these localities, and each of these localities has their own specialty, right? You know where the best vada paos are, for example. You know where to go if you want to actually do furniture shopping, right? So some localities specialize in certain things, other localities specialize in other things. So city is made out of localities, right? Then, of course, in the city, there are roads, freeways, highways, etc., which are connecting the various localities, which enable smooth movement back and forth. Of course, a city is actually physically, if you look at these localities, they are made out of buildings, shops, right, establishments, houses, right, apartments. So these are some infrastructure that are present. So ultimately, what happens in a city? In a city, we make stuff, right? We communicate with each other. We transport stuff, right? These are the kind of things, occupations that take place in the city actually. And ultimately, also we spend a lot of effort, from all the construction that is going on, a lot of maintenance of the infrastructure takes place, right? So this is how a city looks, right? But is this what a city is, right? Is there more to the city? And this is not the life of the city, right? The life of the city is its workforce, essentially us, people. So if I ask you essentially in a city, what is the essence of a city? You would say the essence of the city is people, because people are the ones who make the stuff, who transport the goods, who communicate, who do the things. People are the ones who are maintaining the city as well, for example, right? So this is how a city looks. Now, if I take this analogy and take it to a human body, right, or the body of any organism, for example, you can identify the same parallels, right? So think of the body as a city, right? We have organs, which are the different localities, where each organ specializes in one kind of thing, one kind of task. We have nerves, arteries, veins, which are the highways, freeways, essentially which facilitates movement back and forth. Cells, right, which are the basic infrastructure, right? The buildings, houses, etc. You can think of the cells are the ones that actually make up these localities, etc. Again, what happens in the body? The body makes stuff, right? There is communication, there is transport, exactly analogous to a city. The only difference is that the workforce in a city, in a human body, is basically protein, right? The essence of a city is people. The essence of any organism is protein, ultimately. You must have heard about DNA, because DNA is the molecule of life. All the genetic information, our makeup comes from DNA. But in general, what does DNA code for? DNA doesn't code for eyes or nose or ears. DNA actually codes for proteins. So if there's a person who has blue eyes and a person who has general brown eyes, the difference between these two individuals is because the proteins in our eyes are different. One protein absorbs, a light of a particular color, the other protein is a different color, and therefore you see blue eyes. So blue eyes, for example, are dark brown eyes, these are called phenotypes, right? So these phenotypes come about because proteins are different. So now I come to the essential thing about what are proteins. So proteins are basically large biological molecules, right? They contain many thousands of atoms. And essentially, they carry out all the vital functions that are necessary for life. Just like people carry out all the vital functions of a city, proteins carry out all the vital functions for life in every organism. So I've given you mugshots of 3 proteins, right? The protein on the extreme left performs chemical reactions. This is a protein which has a funny name. It's called cytochrome P450, right? Based on some wavelength of light it absorbs, right? But it performs a very essential function. So, for example, if you've been recently ill and if you've taken crocin, and you find essentially crocin ka effect wears off after 4 hours, right? So there is something happening to the crocin. So this protein is actually acting on crocin. In fact, this protein is a detoxification enzyme. As you go to the city, you must know it's very polluted out there. You breathe in so much of garbage, right? Lots of things go into your body, right? All the stuff also comes out of your body, right? This particular protein actually helps flush out all the stuff in the body. The basic chemical reaction that this protein carries out is called hydroxylation. In very simple terms, it takes stuff that is not soluble in water and makes it soluble in water. It adds a chemical group, it makes it more soluble in water. Your kidneys flush out the garbage out of your system, actually. So P450. The protein in the center is something that we are all familiar with, actually, but you may not recognize it. It's a transporter protein. Can anybody think of a, yeah? It's hemoglobin. It's hemoglobin. That's absolutely right. Hemoglobin. So this is the structure. This is hemoglobin protein. It transports oxygen, carbon dioxide back and forth, right, up your bloodstream, and that is essential for life, for all the activities, etc. The protein on the right, actually there are 2 proteins. There is a protein on the top in orange, and there is a protein, very colorful protein at the bottom. Actually, the protein on the top is a receptor protein. This receptor protein is sitting on a membrane, cell membrane. So think of the cell as like some kind of an egg-shaped thing, right? It has a outer layer, and this particular protein is sitting on the outer layer, right? It takes influences from the outside of the cell and communicates it to the inside of the cell, right? The process of vision, how we see, comes about because of this particular protein. In fact, this particular protein is called rhodopsin. It's present in our eyes, right? And when light hits this particular protein, it changes shape. When it changes shape, the protein at the bottom can come and bind to it, attach to it, right? And based on that, certain reactions take place which enable the process of vision. All this happens very, very fast, milliseconds and faster, right? It's amazing. So the world of proteins is indeed amazing. We know the world around us is biological, so proteins are everywhere around us, right? We know all the different kingdoms of life, right? Some kingdoms you don't even understand very well. Archaea, for example, we still have yet to discover many things. But no matter where you go, you'll find proteins as the essence of that particular life form, right? Inside you, right now, there are 20,000 different proteins. I'm not saying the total number of proteins, basically, but 20,000 different types of proteins right now working to keep you alive at this point. So these are just some of the functions that these proteins perform. By the way, I mean, there can be questions in between, right?
Yes, absolutely.
Yeah, sorry, yeah.
Absolutely. So, yes, you are showing different colors. The only thing is, please repeat the question for the online.
Yeah, I will do that.
There are different colors you are showing. So, basically, our real proteins have different colors, or you are just for demonstrating you are using?
I think that's a very appropriate, absolute question. So this is just a depiction. Sorry. So the question is that I've shown some images, some pictures of proteins. Are the colors actually real as the proteins appear, or are they, just a depiction? So my answer is they are a depiction at the moment. In fact, we will see what this representation means. I've just shown you some pictures, right? But what do these pictures actually mean? They look like squiggly ribbons, right? But is this how all proteins look like? So the answer is a qualified yes. Okay. So I'll give you an example. Suppose some aliens came from some other planet and they started looking at human beings. right? And suppose these aliens had, the way they see and absorb light is very different from the way we do, right? So these aliens have the possibility of seeing through human beings. So they'll see, the outer layer, our hair, etc. They can see the muscles, they can see the skeleton, right? They can see each and every protein, etc. So now, if this alien had to go back and describe human beings, right, he would have to pick a representation, right? So maybe the alien would pick a representation where he shows only a skeleton, right? Because our skeleton is very different from a dog skeleton, for example. So that would be one way to do it, actually. So this is exactly what these representations are. I will now, in fact, go to that particular point right now. I will actually, let's talk about essentially these representations. What do these things, squiggly lines mean, actually? So I'm going to start from very basic scratch. Let's make a protein molecule from scratch, right? And see how this protein molecule is made. What is it composed of? So the basic thing, hopefully everybody knows what an atom is, right? Atom has a nucleus and it has an electron. I mean, if it has a nucleus and only one electron, that's hydrogen atom, which I've shown here in this case. If it has many more electrons, right, then in that case, there are different chemicals. Oxygen, for example, carbon, nitrogen, they have different, more than one electron in that case. So if you take multiple atoms of different types, they can form bonds where the atoms share electrons and they can form covalent bonds. So this is water, right? Well-known molecules. So it's oxygen. So the oxygen is shown in red, and the protons, the hydrogens, are shown in white in this particular case. So there are two bonds that water forms, and this is water molecule. So we come to the basic building block of a protein. This is called an amino acid molecule. Okay, amino acid molecule, right? So what I'm showing you is, for the time being, just ignore the purple R, right? And look at the other components, actually. So you see that there is nitrogen in blue, right? In cyan or greenish, those are carbon atoms. Oxygen is red, and then the protons or the hydrogens are in white. Okay, so this is what an amino acid we call this the amino acid backbone. Okay, and every amino acid actually has the same backbone, this backbone. But what is different about the amino acids are these R groups, the ones in purple. So I've shown you that R group in purple. So these R groups can be large, small, they contain many more atoms, right? And there are 20 of them, 20 basic R groups, which means there are 20 possible amino acids that are possible, but they all have this basic backbone, the same backbone, actually. Okay, so this is one amino acid. How does this connect to the protein? Suppose I take two amino acids like this and I bring them close to each other. Yeah, sorry.
There are only 20 amino acids, or there are more amino acids?
That's a smart.
And whether these 20 are only available with the humans or other?
Yeah. Okay. So in most of the organisms, yeah, sorry. So the question is essentially that there are there only 20 maximum amino acids, right? Are there more? Or are these 20 amino acids only relevant for human beings, right? So this is the question. So the answer is, in most of the organisms, our genes only code for 20 amino acids. There are two additional amino acids. So actually there are 22, but those two are very special cases, actually. So the ones that are explicitly coded and used in most proteins are these 20 amino acids. So just for simplicity, I will just use that. And
just supplement to this.
Yeah.
Chemistry doesn't stop us from creating more than 20 amino acids.
No, it does not, actually. But somehow life has evolved to support itself on these only 20 amino acids.
22 amino acids.
Yeah. Actually, the question that you're raising is a fairly deep question. Why 20, right? Because there are 20, that also tells us how the DNA actually codes for the protein, right? DNA actually codes for these amino acids using combinations of bases, actually. If those combinations change, right, then the number of proteins could also increase. And there are many people in the laboratory who are actually trying to do these kind of things, actually, trying to expand the amino acid alphabet and see what interesting proteins can be formed. But in nature, so far, this is what is the norm in that case. Right. Can I get water?
Last time when we had a similar result,
Yes.
codon.
Yeah, codon, indeed,
Yeah, so
indeed.
there we find that how the DNA is coding these things. That created more curiosity. Why only 20 to us?
Yeah, it's very, yeah. In fact, this is a very common question that we ask when our students come in for a TIFR interview. So these codons are like, they are number of nucleobases, like which are used to code for protein. So it turns out every codon is only 3 base pairs long. So there are 3 base pairs. So think about it as 3, every base can have 4 alphabets, adenine, guanine, cytosine, and thymine. And you take them in combinations of 3 to code for proteins. So the question is why 3? Why not 4? Why not 2? Right? It turns out only when you take 3, and if you have each varying with 4 times, basically, then the combinations are able to accommodate 20 amino acids. But in reality, with these 3 combinations, you can actually go up to 64 amino acids, but only 20 are encoded. So these are part of the mysteries of life. Essentially, if you followed, like, if you know about evolution, evolutionary principles, etc., life has come from very simple forms to certain complex forms, right? And up to this point, we only need 20. As we evolve forward, who knows? Who knows what can happen actually? Anything can happen. But as I said, people in laboratories are exploring precisely these things, how to expand the alphabet. Can the alphabet be expanded? There are again certain organisms where there is more than what is normally observed actually. People are still trying to understand it. Okay, anyways, so again, coming back to amino acids. So these are, you take 2 such amino acids and bring them together. So I brought 2 amino acids close to each other, and you'll notice that I've brought, there is a circle there, right, which says there is a oxygen, there's a nitrogen, and there is a oxygen, right, which is very close to each other in that particular case actually. So if you played Lego, right, in Lego basically there are building blocks, right, that are given to you, and you can take the building blocks and then you assemble it together and you can form different shapes overall. So in amino acids are basically the building blocks of proteins. They're like Lego units which nature uses. So you bring 2 of these amino acids together, and there is a reaction that takes place. It's called hydrolysis. Hydrolysis because the reaction is accompanied by loss of water, right? So you can see if I take that oxygen atom, right, and bring it close to this nitrogen, and let's say, for example, the oxygen and the red is oxygen in that case, right? The red is oxygen. Basically, if it comes close together with the 2 hydrogens, which are the 2 white in this case, then essentially a reaction can form where those can get released in the form of water, right? So you can have a reaction like this where a water is released, right? And there is a bond that is formed between this nitrogen and that carbon. Nitrogen is blue and carbon is cyan in this particular case, right? So this is called a dipeptide. So I've taken 2 peptides, 2 amino acids, and I brought them together and I form a dipeptide. Now you can imagine this reaction taking place ad infinitum. Actually, I bring one more, then I form another bond like that, and so I form something like a polymer chain. So now I've kind of taken away all the atomic details, and I've just put these balls with different colors. So these different colors are the 20 different colors that are possible, and with the 20 different colors, I can form necklaces of different types, right? So this is what a polypeptide chain or a protein chain looks like, right? So this is the basic protein chain. Okay, so how do we get from this to that squiggly thing that I showed you, those pictures that I showed you? Okay, now some very interesting things happen actually, and this is fundamental to our discussion about the protein universe and artificial intelligence and everything. So something called protein folding happens. Okay, so suppose I take this particular chain and I drop it in water, right? What will happen? So this chain, it turns out, depending on those R groups and the colors, right, can take a variety of different shapes and arrangements. Right? So these are the standard ones. For example, in this particular case, and I've here completely in my computer, I've told it don't show the R groups because otherwise it will become very complicated. So I'm only showing the backbone of the amino acids, and you can see essentially there is a very nice periodic arrangement actually where they are all like in planar, right, and they're stacked to each other. So if I put this in my computer has the ability to quickly just make this into some kind of a cartoon representation, so I can quickly identify it. You can program this into the computer. Like if, for example, all these things come so close to each other, or like you have like these kind of arrangements, then instead of showing that, which is on the top show what is on the bottom, right? So essentially, this is what is called a beta sheet. Okay, so this is called a sheet-like arrangement. This is one possible arrangement if I take these amino acids and drop it into solution. And actually, you'll notice essentially that, I mean, I'll use the pointer in this case. So there are these sheets, right? But essentially, you see this more disordered segment, actually, right? This disordered segment is called a coil, a random coil, right? So if you cannot assign it to a particular sheet, then you put it as a random coil. Another arrangement is this one, also very interesting. Actually, if I take again things of certain colors, right? These particular backbones can arrange themselves into this kind of a helical form, a coil, right? Actually, it's a confusing thing. I said I'm only showing the backbone. I'm not showing the side chain R groups in this case. I'll come to that in a second. Actually. Just for clarity, again, remember that alien who comes to our planet and he wants to understand, basically. So he doesn't want to see everything, and like if you show everything, it's going to be very difficult to understand. So essentially, you're just abstracting this. Anyway, so the computer will draw this ribbon for me. When I have this ribbon, I know that my backbone is arranged in this helical form, right? When I have the sheet, I know essentially my backbone is all planar like this, and it's arranged like that. Here is an actual protein. Okay, this is a protein called a DNA polymerase. It's actually maintaining our genome inside our body, right? It replicates DNA, right? And it's shaped like a hand, right? And I will come to the significance of that in a second. But look at this particular polymerase. Here I've not done that. I've shown you the polymerase in all its glory, all atoms, right? And you can see how it looks like a khichdi. Broadly, you can see like a shape, right? But you can see all the atoms, right? But if I put it in this representation that the computer has drawn below, then it looks like this. Okay, so now you understand those three pictures, those three things that I showed you. Now again, the colors here are also by choice, just to make it colorful, make it attractive. We can simulate it on the computer, but right now this is just, even this can be obtained by experiment. We'll come to that as well. In fact, the topic is how do you get this structure, right? How do you get this structure in that? But you can see what this representation is. I hope it is very clear. When I show you a protein and tell you that this is a protein, what is being shown to you, hopefully, right? If there are any questions, we can take it at this point. There are no, sorry, man, it's such a tough task. Yeah, sorry. So the question was, is there a limit to how long this polypeptide chain can become? Yeah, so I don't see any physical, limit to this actually. So for example, it depends on the protein that you have. For example, in a body, the largest protein is a protein called titin. This is in our muscles, right? So when you exercise, we see our muscles growing, right? And you can see that growth, right? I mean, it's physically, you can see that growth. It's because this protein has this elasticity associated with it, where it can actually become like, longer or shorter. This titin has 35,000 amino acids. 35,000, right? Some of the smallest proteins of the polypeptide chains actually have about 30, 40 amino acids, right? Functional. And a moderate sized protein would be somewhere between 200 to 500 amino acids overall. So this is the scope, but you can go as much as 35,000 if you want. Yeah. Good. So this is the question again. So the question is this. So the question is essentially what determines how long a particular protein should be, right? So I would say mostly one word answer to that is function, right? What is the function that is desired of the particular protein that leads to that, and essentially the function itself is related to the structure of the protein, right? And whatever number of amino acids are needed for the protein to assume a particular structure is what nature would give you. Right? Okay, is that clear actually? Okay, in fact, that's what we are coming to next. Actually, structure. So the function of a particular protein, right, indeed is connected to its structure, right? It's connected also to something called dynamics, because something which is just sitting in one place and not doing anything is not going to do anything, right? I mean, sitting in one place and not moving is not going to be able to do anything. So it has to respond to the solution, to the temperature, etc., and that is called dynamics, right? And once structure plus dynamics come together, then the protein performs its function, right? So this is a critical thing. So the structure of a protein is the basic thing. So a protein assumes a particular structure, and based on its structure, you can actually tell what the protein is up to, right? So again, going back to our example of the signaling protein, right, where I showed you the receptor. Now I've just shown it in a different color, if you notice, right? So for example, the protein is sitting, now I'm just use the pointer again. So the protein is situated in a membrane bilayer, right? So this is the boundary of the cell, right? And this particular protein is sitting inside the membrane layer. So notice if I have to make a protein which actually penetrates the membrane bilayer, right? It has to have a particular shape. It should have a screw-like thing, right? Remember, if you hammer something in or whatever it is, a screw-like, topology actually helps you penetrate, for example. So this particular protein, if you notice, actually has all these coils, right? So it's coiled, so it can penetrate the membrane in that case. So this is a very characteristic structure of a protein like this. It's called a transmembrane protein. It's called a transmembrane helical bundle, right? So many proteins which are actually doing this kind of signaling, right, are like this, shape like this. Yes.
This type of protein is used by COVID to penetrate in our body cells?
Yeah. So the question.
Curiosity.
Yes. So the
curiosity.
right. So the question is this kind of a protein also responsible for penetration of COVID into human cells? So the answer is yes. There is a receptor protein in our body which is performing some other function, right? It's called ACE2 receptor, and the SARS protein actually uses this receptor as a target. It actually attaches to this receptor and then penetrates the membrane. Right. So the second protein in the middle is the DNA polymerase, the guy that I showed you before. But this time the polymerase, I've shown the polymerase which is bound to DNA itself. You can see that colored helices, right? The red and the yellow, right? So this is why the polymerase is shaped like a hand. It's shaped like a hand so that it can actually grip the DNA. So the DNA comes and sits in the groove, right? And by motions of the thumb and the fingers, this particular polymerase can actually add nucleobases to a DNA. It's amazing, actually. It's just like a hand which is adding nucleobases to DNA, actually, right? It's a fantastic reaction by itself, but the shape of a hand is responsible for actually accommodating the DNA in the groove. There's another thing which is also important. This particular protein has a lot of charges in the way the R groups are, so that DNA which is negatively charged can actually attach to this particular protein in a particular, right? So this architecture is very important. The structure is very important, right? And again, we talked about yes.
DNA will also be represented as molecule. Then the protein attaches to the DNA.
Yeah.
DNA also like a set of molecules or
so yeah. So the question is DNA also a molecule, or is it collection of molecules, or what is it? Right. So DNA by itself is it's a large biomolecule. DNA by itself, right? But DNA is composed of these units called nucleobases, right? So adenine, guanine, cytosine, thymine. These are the units of DNA, but they all have the same architecture. There is a backbone of DNA which is stretching, and then each of the backbone has this hanging nucleobase, each strand of DNA, and this pairs with a complementary strand. So the DNA by itself is a molecule. It's a molecule which is actually coming together. Yeah.
why is DNA negatively charged?
Why is DNA negatively charged? Yeah, that's a deeper question than say. So I would say that the negative charge of DNA. So essentially, the DNA carries genetic information, as you know, and it's sort of like a repeating polymer, right? Overall, your cellular machinery needs to manipulate DNA. Manipulate DNA in the following sense: that at inactive times you want to store the DNA in a very efficient way, right? Right now, for example, in our body, etc., basically, every cell you have about a meter's worth of DNA or something like that, right? but the cell is so tiny. How are you going to store the DNA? So you need, no. So essentially, exactly. So you know more than that. Basically, so there are proteins, and the DNA is wrapped around the proteins tightly. Okay. So what happens is the negatively charged backbone, but then so you need to really wrap the DNA tight. But at other times, when you actually want to take the DNA and read it and create proteins, for example, you want to stretch the DNA out and make it rigid. Okay. So what happens is the cell uses this wonderful mechanism where if you have negatively charged backbone of the DNA, because there are negative charges on the backbone, the DNA actually bends quite a bit because all the negative charges want to go away from each other, right? So it bends. But if I take the solution and put in the solution something like magnesium ions, which are positively charged, those magnesium ions go to the backbone and they sit on the backbone and they neutralize those negative charges. Now, then the DNA, because all the negative charges are gone, the DNA becomes rigid. It's stretched out. So just by controlling ions in your environment, right, you're able to kind of control the architecture of DNA. Yes.
If we could take an online question.
Yes.
So, if you're talking about the medium, is the structure of the protein having an effect on the surrounding, especially pH, temperature, and medium?
Effect of the protein.
Effect of the protein on that, or does the pH, temperature, and the medium affect how the protein structure?
Absolutely, absolutely. So very valid question, and yes, indeed, the pH, temperature, all of these factors are important. In fact, we will see again very shortly, essentially how when you talk about the protein folding paradigm, right, how these factors come into play. So short answer is yes, they do impact. Okay, so yeah, again, detoxification, just coming to the last particular protein again, right? So this I told you performs an important chemical reaction, right? For it to perform a chemical reaction, there has to be, so a chemical reaction is, think of it like cooking. If you, essentially cooking is a chemical reaction, right? So for cooking, you need a pot, right? And you need a place where you have to put the ingredients together basically and cook the ingredients. So chemical reaction, there is a transformation that takes place, and right, so the protein needs to have a site where the chemical reaction can take place. So this particular protein, you can see that there is a green ball that I've noticed put in the middle center actually. So that is called the active site of this protein. And this active site, it has the right apparatus where it can carry out that particular chemical reaction. So again, just by looking at the architecture of the protein and its active site, you can tell a lot about what the protein does. More importantly, right, if you're designing drugs, right, and trying to change the function of a particular protein, something that is hyperactive, you want to tone it down, you need to know the structure of the protein to know essentially how to design those drugs. Many drugs actually are discovered empirically, right? Dada, Dadi, Nana, Nani have been using something and then chal raha hai, and everybody knows that actually. But scientifically, you would like to understand essentially what are the targets, what are these things actually impacting ultimately. And if you look at it, most of the targets for the drugs that are designed, etc., are all protein targets. There are also nucleic acid targets for cancers, etc. This is important, right? But protein targets are the majority of targets for the drug. In fact, 50% of the drugs that are there in the market actually are targeting the signaling protein that I showed you. One form of the signaling protein. Blood pressure medicine, for example, they target ion channel receptors. So your calcium ions, etc., they flow out basically. So if your blood vessels have to open wider or not open wide, essentially, then these are the receptors that you have to target overall. So if I know the structure of the protein, then I can actually design drugs much more intelligently, right? So structure of the protein is critical. But there are 20,000 proteins inside your body right now at this point. How can you figure out the structures of every one of these proteins? How easy is it for me to figure out the structure of the protein? I'm just showing you these pictures. How easy was it for the scientists to come up with the structure? Right? Not very easy at all. In fact, the first structure was discovered in 1960s. Right? In fact, the Nobel Prize was given in 1950s. And for that, they actually got the Nobel Prize for that. And structures of this particular protein, the signaling protein that I've shown you, were discovered only in 2013, for which the 2013 Nobel Prize was given to that particular person. Determining the structure of protein is a very complicated thing depending on the protein at hand, right? So that's where we are going now at this point. So what are the tools that we have for protein structure determination? So historically, this has been an experimental field, right? you want to determine the structure of something. I want to know essentially how a human being looks. What do? I take a photograph of the human being. So there are two methods by which you can actually take these photographs. In fact, one of them is X-ray crystallography, right? Both techniques are actually familiar to us, right? X-ray, we always get X-rays of our bodies done basically. So X-rays are passing through us basically, and then they are able to show you inside your skeleton, etc. And the second one is called NMR spectroscopy, which is a variation of MRI, if you want to think about broadly, right? In X-ray crystallography, you bombard, you first of all have to grow protein crystals. So the requirement for X-ray crystallography is that the protein solution has to become crystal. Like you might have seen salt or sugar crystals, right? So protein, when you take the solution, you have to as you cool it very slowly, it has to form crystals. And the bigger those crystals are, the better the X-ray crystallography, right? You hit it with X-ray, and then you get a diffraction pattern like what's shown up here in the second thing. This is a diffraction pattern. Like these look like random spots, but people have figured out how to take these spots and invert it to actually find the corresponding distances between what is the chemical composition of these spots, right? And what are the distances between the groups. So people have figured it out, and ultimately you get a protein structure, right? And there is some process of iterative refinement. Once you get the structure, you see whether everything is fitting or not, and then you go back and check the diffraction pattern, etc. And then you can get the structure of the protein. NMR spectroscopy is also very, it's slightly different in the sense that the protein is in solution. Instead of actually growing crystals of the protein, you can actually take the protein, put it in solution. And in the solution, you can have a magnetic field, right? And then you hit it with microwave light. And then at the end, essentially, again, you get like some kind of a spectrum, which tells you something about the chemical composition. And, so again, I'm not getting into the technical details of this, right? But ultimately, at the end, people have figured out how to use these patterns, spectra, etc., to figure out what the chemical composition is. Okay. So what is the catch actually? This is great. How are we doing on time actually?
Almost 45 minutes.
Yeah.
You need to play your game.
Yeah, I need to play the game as well. Right. Anyway, so that's great actually. But what is the catch? At least I'll tell you what the issue is in this case. So for a single protein, this process, right, can take months or even years, right? Because for you to, for example, you take a protein solution and the protein doesn't crystallize. You don't know why it is not crystallizing, so you have to figure out, you have to play around with the composition, you have to understand what's happening chemically in the situation, right? Maybe the protein also very unwieldy, it's very shaky, it's moving around a lot. You have to stabilize it, you have to add things, you have to subtract things. It can take months to years to figure that out, first of all, right? And then after that, you can actually figure out the structure, but the equipment that you use is also in crores, for example, for you to get that done. So far, since X-ray crystallography has been done and NMR has been, in action, we have something like about 0.2 million, like 200,000 structures have been figured out so far for proteins. 200,000 proteins have been figured out, right? Yes.
Could a protein fold into a particular shape every time in a given condition? Would the shape change even if the conditions are the same?
Yeah. So so the answer is if the conditions are the same, then the protein will fold to the same structure. There are certain classes of proteins which don't show that, right? There's special proteins which don't have a particular structure, right? That's true, actually. But most proteins which are structured proteins will fold to the same shape overall. If that basis was not there, then this structure determination is meaningless, right? So, but I just want to emphasize 200,000 proteins have been throughout so far, right? Not only 200,000 proteins have been figured out at this point, right? Even in terms of a human body, 10% of the proteins inside a human body, we know the structures of those proteins. 10%, 90% we don't know, or at least didn't know till now. So now we are coming exactly to this particular question actually. So these are experimental methods, right? So can something be done? Because if we go at this pace, it's going to be impossible for us to cover the protein universe. Because I told you, right, there are a million species around, right? Million species around. The proteins in different species are different. Human beings have 20,000 proteins. Rice has somewhere above 35,000 proteins. Rice, right? There's a parasite basically which causes urinary tract infection basically, and that has about 60,000 proteins. So it's not like, you get, if you go to more complicated, if you think of ourselves as more complicated than that parasite which is causing urinary tract infection, the number of proteins in our body is less than that in that particular different types of proteins, I'm saying, right? Is less. So how are you going to figure this problem out, right? So you need something more. So we have to go back to the basics of essentially where does this structure come from, right? What is the underlying physics or chemistry associated with that? So Anfinsen in the 60s actually figured out a very interesting point to this problem. Actually, he was working with proteins, and what he did was he did this interesting experiment. He took a particular protein which he knew what its function was. It was doing some kind of a chemical reaction, right? He applied conditions, right, where the protein actually unfolded. You can do this in different ways. I've shown you here. I'm showing temperature, right? You take the protein and you apply high temperature, the protein will unfold. In that case, right? When you cook eggs, for example, the same thing happens, right? But then now once the protein unfolds, you can then cool it back, right? And ask essentially, is it going to go back to the same structure? So the way Anfinsen figured it out is the particular protein is doing a job, right? I told you to do its job, it needs to have a particular structure, right? So if I unfold a protein and then I refold it back, is it going to continue doing its job? Is the question that he asked, right? So he found essentially very startlingly that his chemical reaction was intact even if he was able to put the protein back. So this led to a very interesting hypothesis. So the idea was that if you take a particular protein chain and just unfold it and make it a linear chain, the protein by its own natural forces, following the laws of physics and chemistry, will go back to only one structure. See, this is very not intuitive at all, actually. If you, for example, played around with your telephone wires, you take a telephone and you kind of hold basically, and then suddenly, I don't know how this thing folds into this random thing actually. But every time that randomness it appears very different. But in this case, what you're saying is the protein is going back to the same fold, right? If you're actually leaving the thing. So which means that the sequence, the colors, the way the colors of the amino acids are arranged in the beads, that actually determines what the final structure is going to be. This is called the sequence to structure relationship. It's one of the primary driving, forces in structural biology. So these are very interesting questions. Why is this unique 3D structure attained, right? So initially people did some back of the envelope calculation. You take every particular amino acid and you ask, essentially, for example, if I let each amino acid explore some, particular twist or whatever it is, right? How long will it take for the protein to reach this one final structure? Turns out that back of the envelope calculation tells you it will take you longer than the age of the universe, right? But that doesn't happen in our body, right? In our body, it has to happen in milliseconds, seconds at the most for some processes. So how does the protein do it? How does it actually do it? So now we actually understand a little bit about the physics. So that assumption that was done, saying that random movement, that assumes that it's something like imagine, for example, that I scatter some beads on this very flat landscape, and I ask it essentially whether it's going to form a particular pattern. It may not form a pattern, right? But instead of this very flat landscape, suppose I had like a well with shapes, some mold, right? And I drop all the beads, and all the beads will accumulate in a particular point, and then a particular pattern will be formed. So this is what we call the energy landscape of the protein, right? And this energy landscape is responsible for driving the forces. So we think we understand these driving forces now, definitely more than that, but really has that information translated into us predicting the structure from the sequence. So before we go forward, right, I would like to play this protein folding game, which will kind of emphasize essentially what these rules are. What are these basic rules that we figured out? So you guys can also see how a particular protein can fold to a particular shape. The same thing that I just mentioned, flat landscape versus this actually. So maybe let's try to so I need 20 volunteers. I don't know. We have more than 20 now, right? I need 20 volunteers.
So do you want to just stop the sharing?
Yeah.
Because that way we can see what's happening. Because otherwise we'll be stop sharing.
Okay. So I'm going to stop sharing.
I mean, unless you want to just give the You don't need anything more than this slide, right?
I need to just make, so before we set up, so there is a setup.
Explain the game and then you can.
Okay. So the setup is very simple actually. Here, okay, I have here with me, like, 20 cards. Okay. So these 20 cards are representing the different kinds of amino acids, right? So you can classify the amino acids. I told you there are 20 colors, right? But those 20 colors can also be classified into certain types. Okay. So what are those types? So I have created some white cards, right, which are called hydrophobic groups. Hydrophobic means basically that these particular amino acids don't like water, right? They want to run away as far away from water as possible, right? Oil, for example, right? Oil doesn't like water basically, so they don't mix, right? So similarly, in this particular case, now the pink cards are hydrophilic. These amino acids like milk, milk mixes. That's right. Oh, sorry. Yeah, I thought you meant hydrophobic. Yeah, right. Sure. So hydrophilic loves water, right? So essentially, again, there's a mixing. Then there are amino acids which have positive charges, right? There are amino acids which have negative charges, right? So positive and negative. So electrostatic. Positive and negatives actually attract to each other, right? But so we'll classify them as blue and red. And then there are sulfur-containing amino acids. Okay, there are only 2 of them. So they are very interesting also because there is a sulfur bonds can form in this particular amino acids, right? So we're going to take these groups, right? And I'm going to just put some simple rules. Okay, I'm not saying that these rules are actually they go in this hierarchy because it depends a lot on how many of them there are. Suppose there are a lot of hydrophobic groups, then that particular thing will drive the protein folding more, right? But we are going to simply keep 3 rules at this point, right? So we will first of all form a chain of these volunteers. So each volunteer is going to represent an amino acid, right? We are all going to hold up these cards, right? And we're going to lock arms, right? And we're going to stand whatever end to end, right? Now, so this is the linear chain, right? And imagine that if you put this chain in water, maybe I'll have these kids running around. These kids are water molecules, right? Water molecules will run around. So the people who are hydrophobic, they're supposed to run away from the kids. The kids should not be able to catch them, right? The people who are hydrophilic, they will try to catch the kids. The kids have to avoid those guys and run after the hydrophobic guys, right? Then essentially the charge group. If somebody is positively charged and there is somebody else who's negatively charged, you have to find them and try to connect to them, right? So you have to come as close to them as possible. And finally, if you have 2 guys basically who have sulfur, then you are supposed to come really close to each other because there is a sulfur-sulfur bond that can be formed. So these are the simple rules that we're going to use, right? And so the idea is I start with a linear chain. I'm going to randomly hand out these cards, right? I don't know which group is going to go where. So this linear chain will form some protein chain. And then we'll see essentially what the protein will fold into. Who knows what it will fold into? Maybe it will be khichdi. Who knows? But let's see. So the rules are very simple. So first, the sulfur bonds get priority, right? You have to allow them to move first, right? Then the ionic bond, which is positive and negative, right? And then the hydrophobic collapse. Then the children have to attack the people basically who are hydrophobic, right? And the hydrophobic guys have to run away from them. And at the same time, you have to avoid hydrophilic guys, right? So this is what the game is going to be. So can we try it out? So I have the cards. Actually, I don't know.
20 people. I don't know how we're going to do it here. We may have to once people come
No, no.
up here, we'll make some sense.
Yeah, I have a suggestion actually. So if the people who are not participating can line the borders, then we can use the entire room actually. That may be one possibility.
You need that thing.
This thing. Yeah, we just leave it. It doesn't matter actually. I can.
Otherwise, nothing is visible for the online audience. You're on TV right now. What people are saying that as long as it's on screen, that we can perhaps we could just make the whole thing here.
Okay. So we'll minimize this. You don't have. You can. I can unshare. Yeah.
wants to play? You want to play? Here's one.
No. You guys are water. I need water molecules. Water molecules are really fast. They're fast compared to the amino acids, right? So we want amino acids to be the slow, sluggish guys.
Oh, okay. Those are the separators. Sorry. Just make sure your thing has a 3-digit code on it.
See, each 3-digit code is a particular amino acid, right? So I'm not mentioning the names, but there is a 3-letter code which simply tells you. Yeah. All you know is it's hydrophobic, hydrophilic, right? Yeah. Then you have to escape from them. Actually, if they catch you, they catch you. There's nothing. You try to run away from them. You're faster than them, right? They shouldn't be able to catch you. See, they're all locked down. It's going to be difficult from the. Yeah, they can't. Yeah. No. These 3 digits, they're just names. Like, for example, this is a code for lysine. That's why you say LYS.
Okay.
We just have to touch the hydrophobic.
One more.
Yeah. You see, you try to go as close to the hydrophobics as possible. Right? You try to stay there, but the hydrophobics will try to go away from each other, and the hydrophilics will. You have to stay as far away from the hydrophilic,
And as
close to the hydrophobic as possible. Try that. Okay. So now what we'll have to do is, so we have 20, right? Okay. So let us start here. Let the first person just you can. Why don't you stand here? Right. Okay. Second person, lock arms. So then lock arms. There. Right. So now we have unity in strength or something like that. Let us.
And you see, we are part of the theater festival.
Yeah. And then yeah, lock arms.
Can we just get this lysine also here? It's okay, right?
Yeah, of course. Yeah. Okay. I need some more waters actually. Some smaller members.
Oh, I can.
No, you're too big. Okay, Arnab. With his excitement, I think. Yeah, come, please. My wife could also qualify actually, but she's doing some busy jobs. Okay, so we have only 3 waters. A little bit few more.
Yeah, 4 waters.
Oh, 4 waters. 4 waters. ठीक है. Huh? Okay, she's.
No, it's okay. You can take something.
I can take some pictures.
Okay, fine. We'll get.
Achha, okay, guys. So everybody is logged on. Let me just make sure that everybody is logged on. Okay, very good. So the locking of the arms is essential because then you don't want full freedom to move. Right? That's how the amino acids are. They can't freely just move. Okay. So right. So we have lysines, positive hydrophobics, hydrophilic, negative charge, cysteines. Okay, cysteine. Sir, can you actually come here and you exchange? I'm now designing the protein. So that there is थोड़ा cheating हो रहा है. Actually, sir, why don't you come? Sorry to you come here. I'll make it a little bit more complicated. Yeah. Now you lock arms with her. Actually. Right. Okay, very good. Okay, so now I'm going to just read out the rules, and you guys are going to obey the rules according to the order in which I'm going to read out. Right. So the first rule is that sulfur groups are supposed to come together. So this is the protein which is in solution. Now it has to fold. Right. So the first thing that has to do is sulfur groups have to come together. Can sulfur groups come together?
Yellow.
You identify. No. You have to come together, but the entire chain has to move, right? There you go. So sulfur groups are together. No leaving hands. No leaving hands. Turn towards her. Right. That's it. Okay. So this is the first. So this protein first finds a sulfur group, and now it's formed a disulfide bond, and you can see that the shape has now changed to this kind of a thing now, right? Okay. So now this has formed a constraint now. Now, rest of the movement has to happen without breaking this. Okay. So now all the positively charged guys find negatively charged guys and try to get close to each other.
Get close to
Positive charge. Positive charge. Negative charge. So have you formed a figure 8 now? Infinity one gap actually. Okay. Now I'm going to add, right, hydrophobic collapse at this point. And hydrophilic guys have to go out and hydrophobic guys have to go in. Right? Attack the hydrophobic. And hydrophilic guys catch them. Yeah, Arnav is.
I would mind the height. Yeah, I can.
Yes, fine. No, these two waters are not caught yet. Hydrophilic guys, what are you doing?
You have to catch the hydrophilic. Where is hydrophilic? Catch hydrophilic. पकड़ लो. बेटा पकड़ लो.
No, you can't go that far. You have to catch the hydrophobic guys, remember.
The hydrophilic. Hydrophilic को पकड़.
Correct. You got a hydrophilic.
इसको कोई पकड़ा नहीं. Right. Oh, hydrophilic. Any free hydrophilic? All hydrophobic. Then go inside further, deeper. Right. एक पकड़ लिया. Okay, good. Huh? Yeah. You have to go in front of them actually. Otherwise it will.
No, it's okay.
All right, guys. Okay. Hydrophilic, yeah, right, very good. Okay, guys. So have we still maintained the figure 8? No. The cysteines are still there. They're close by. You guys managed it. Fantastic. Right. So it's not exactly a figure 8 right now, but it's formed a collapse structure now, right? So initially you started with a wide structure and then you formed a collapse structure, right? The collapse structure has some architecture at this point. So this is a protein folding thing.
How long does the body take to do?
Milliseconds. Milliseconds or even sometimes faster. The fastest folders are on microsecond time scale. Yeah, the fastest folders.
And that too within inside the cell.
Inside the cell. Yeah, that's a crowded environment too. Yeah, thank you. Yeah. Yeah. Thank you guys. Thank you for participating and being sporting actually. Actually, बड़े से actually. So guys, if you guys are interested in this, think about designing your own protein sequence, right? These things can be done by everybody. I can even send these 20 amino acids or whatever slides. If you want to try it out, in a bigger crowd with bigger room, basically it's an interesting game to play. And you ask essentially what kind of patterns can arise. Can you really choose the amino acids in the correct way so that you can actually form that helix, right? Can you really form that sheet, right? So these are some very interesting exercises that can be carried out just on your own if you're interested in this thing actually. And you don't have to do it with people, by the way. You can also form these beads and strings, and you can actually have these blocks, and then you can try to arrange it by hand and see what kind of patterns it forms, etc. Actually, so this is the basic protein folding problem. So see, this is very different in spirit to the experimental methods to determine the structure, right? Experiment mein kya hai? You directly crystallize and you shine light and you see what the structure is. But here you're starting from basics and asking what kind of patterns can form at the end. So it's a little bit more, fundamental approach towards understanding. It requires a lot of understanding. Okay. So yes. So I'll now.
Present study,
Yeah.
which we do generally using AI, which you will touch right now.
Yes.
Do the same type of thing.
So that's what I'm coming to exactly. So please just give me 1 second. We'll just sit down again, and I'll just now make a few comments. But hopefully you guys got the idea, right, in terms of how the folding happens, what there are rules to the folding. Oh, your questions? Okay, we'll take the questions actually. So do you want to ask?
Where you understand where you were standing before?
Yeah. Okay. If you don't mind, because Arnab uncle is really creating problems for me and scolding me actually at this point because of the time. So we will just
my suggestion is be almost an hour. The chai will get cold. So what we will do is we'll try and finish off a part, have chai, and then we come back. And after the chai break, the online audience doesn't stay.
During chai, I will talk to you about this anyways.
Okay. Yeah. So let's go ahead.
Okay. So you guys got the general rules, right? So now I can do exactly what we did on the computer, right? In the computer, I can take a chain, and because I know what the rules are, I can ask the protein to fold by itself. The computer will already move the amino acids and then fold it, and it will tell me whether this is a good arrangement or not, right? Is this a stable arrangement or not? This is what happens also in real life. The protein folds. It's not like, we fold it, right? But when we fold it, after you fold, you see around you each other, and you ask yourself, is this the best approach, best I can do? If this is not the best, so the protein can also unfold back again and then refold into a better configuration. So in nature, there is no one-time hit, right? You can do it again and again. And oftentimes, different parts of the proteins may, slightly open up to accommodate more things and then do it, etc., until it reaches that optimal shape where it can actually carry out its function, right? So this is the essentials of protein folding. So I can use the computer to do this. So the question that's been around for now in the field for quite some time is, these experimental techniques are great, but they're very slow. They're very expensive. If I can do it on the computer, starting from the sequence, I can do a lot more, right? So this is the essence of, I'm sorry. This is the essence of a particular type of competition that has been created in the field called Critical Assessment of Protein Structure Prediction, CASP. Since 1994, CASP has been carrying out this biennial, competition. Every 2 years, all the best computer people basically who are modeling the protein, they get together and they check how good is their method to get to the final structure from the sequence. Okay. And this has been happening since 1994, right? And the idea is very simple. The CASP organizers contact a lot of experimentalists and they find out, which protein structures are going to be solved in the next few months, right? So they know that maybe, let's say there are 100 structures of varying difficulty, simple to difficult. What is simple? Simple means, there are many examples of such proteins, for example. And difficult, nobody knows. There is no such example of a protein, so God knows whether the structure will even be obtained in that case. So easy to difficult targets are identified. The experimentalists are contacted beforehand. They are told not to release the structures to public. So these structures will not be published. They will be held back. All these groups will then be given these targets. The sequence of the protein will be given to them. The amino acid chains will be given to them. They will be given exactly 3 weeks. In 3 weeks, they have to submit a final submission, which says essentially, this is the final structure. It's like playing a video game. ultimately. And so this has been going on since 1994, a very interesting competition, right? CAS 14, which was held in 2020, featured, for example, 97 groups from 19 countries around the world, right? And there are about 215 methods that were applied to predict the structure on about 100 experimental targets, right? Essentially, the results are what I'm showing you on the left-hand side, right? This graph. This graph is crazy. Okay, it has got a lot of detail. So I'm going to go through this in just show it to you in the simplest possible things actually. So what is being shown on the y-axis? Sorry. You can see that there are scores from 0 to 100, right? More is better. 100 is the best. If you have a perfect match with experiments, you get 100. 0 is you have no match with experiments, right? And on the x-axis, you're seeing targets, easy targets and difficult targets, right? So anything that is on the left-hand side are easy targets, easily that can be solved by the groups, and difficult targets which are very, very difficult to solve. So what you can see is some lines, right? So what are those lines? Those lines are results from the CAS competition since 1994, different colors. And you can see some dots, some data right here in this. I just want to go there. It's easier for me. These dots here, right? These are the results from the CAS 14 competition, which was held in 2020. Okay, so you can see, let's take this line, for example. This is the first competition, for example. You can see that the groups do very well. They're very close to the final prediction when the target is easy. But look at how bad it gets when the target becomes difficult, right? And you can see gradually this thing has been improving over the years. Over the years, it's been improving. But there's a big dramatic jump here in this year, right? But in this year also, you're slightly close to 60%.
What is that year, sir?
This year is the last 2018 CAS. There's a reason for this also.
Deep learning has come.
So here, AI-based methods have started here. So you can see this thing for difficult targets has jumped to here with AI-based methods, right? And this year, you have more than 80%. Most of it is actually higher than 90%. Higher than 90% is as good as experiment. As good as experiment. In fact, I'll tell you something even more astounding compared to that. Not only is it as good as experiment, it's actually better than experiment. I'll tell you why. So this is the benchmark that you have, and you see this dotted line here. The dotted line is that they have removed the data from only one group. One of the competitors, they removed the data from that group, and they showed essentially what the rest of the field is doing. So the rest of the field is here. And this improvement is all because of only one group. Only one group which has given the competition, basically. And this group is AlphaFold by DeepMind. This is the group of DeepMind, and they use a program called AlphaFold. So AlphaFold has blown this competition out of the water, right? It's most of its hits, even for the difficult ones, are above 90%, which is experimental quality or better, right? See, experiment is your measure. आपका पैमाना है experiment, right? So you're comparing everything against experiment, right? But this thing is so good that it is becoming the paimana to experiment right now. Why is that? So because people did this, right? People have actually taken different experimental methods, and different experimental methods themselves have resolution issues. Some are very high resolution pictures, some are low resolution pictures, etc. Right? Previously, this was never an issue. Low resolution picture agar experience aa gaya, to be it's much higher than competition. But now, even the low resolution, as the resolution of the experiment increases, right? You can see that the score actually goes down. What this is saying is that if your score is lower, it's not because the computational method is bad, it's because the experimental reference is bad. It depends on that, right? That is amazing. You completely inverted the thing that you're actually comparing and competing in this case. This is a structure of a particular protein domain, which is particularly difficult. And the cyan. So what I've done is I've superimposed these two structures together, right? So superimposition of the two structures tell you that they are almost identical. There are small differences, of course, right? But they're almost spot on.
Which university has developed AlphaFold?
Yeah. So it's not a university at all. It's actually a company. called DeepMind. This is a affiliation of Google. Google has a particular arm branch in the UK. Essentially, this company is called DeepMind. It's headed by this guy called Demis Habasis. And Demis Habasis and John Jumper, who's the primary programmer, basically, they developed this code called AlphaFold. Even here, this dramatic improvement that you got was because of AlphaFold. Right? This is AlphaFold actually giving you this kind of a big jump. And now the version of AlphaFold that they've released is even better. Right? And this is, I mean, you can't do better than that. Right? That's exactly what I'm going to talk about. Maybe after the chai. Okay, I'll tell you one. I just want to show you this one slide. After that, we'll just stop. Now, this is just amazing, actually, in my opinion. So what happened is, I told you that before the competition, these guys contacted experimental groups, right? And they told them basically that they should actually crystallize or solve the structures and don't release it to the public. Now, what happened is, for a small case, number of cases, about 10 or 11 structures, the experiments themselves failed. They could not get the structure because the proteins were so difficult that they could not get the structure. Neither X-rays worked, X-ray crystallography worked, nor NMR worked. Even emerging methods like cryo-EM did not work in that case. So now what the organizers did now is they gave those sequences to AlphaFold anyways, without any experimental structure. And AlphaFold predicted the structure. And these experimentalists then went back to the labs. They took the predicted structure. Because of the prediction, they found out what was the issue. And they solved the structure after that. And these are emails that those guys sent to CAST. So look at the first email. The first email is the most solid email, but you can see. The model you sent me from the leading group, right, worked for molecular replacement. And we finally solved the structure of, it's not Mr. SAD, it's MRSAD. I am still astonished that the human expert model worked, while none of the server models we tried did, as they were rather similar. And this is Henning Kidder. Okay, so he's quite shocked that a human, type of model, the group actually was able to solve it. Second one, I cannot overstate my excitement at the fact that Marcus Hartmann, a student in the group, solved the structure of AF1503 by molecular replacement with the models of group G427. This is again AlphaFold, Andre Lupa. And look at the last one. We are shocked, stunned by the quality of the model. You would not believe how much effort we have put into getting this structure. Years of work, both cryo-EM and crystallography. I mean, this is really shocking. They spent years trying to solve the structure experimentally. All the years of effort did nothing. AlphaFold was able to predict it. And how much time does AlphaFold take to get these structures? Sorry, just 1 second, just one punchline. 15 minutes for the smallest proteins and less than half a day for the biggest protein. 15 minutes for half a day, right? They were able to just blow this competition out of the water overall. So this is what artificial intelligence. Yes, please.
What they've said is that their researchers have come up with a model, AI model, which can predict the protein folding 60 times faster than the current state-of-the-art model. So it just got 4 days ago. So I think this talk is very timely.
Yeah. No, so it's artificial intelligence methods are really making big inroads, and people are now optimizing even beyond what AlphaFold is doing right now. And AlphaFold itself and DeepMind itself is facing stiff competition from many emerging players, actually. But one has to give it to the people who did it first. So in this particular case, they are the ones who actually managed to really, yeah.
An online question from Swapnil.
Yes.
Even if we determine protein structure using computerized methods, we will still have to confirm by X-ray crystallography studies, which is the gold standard. So what's the advantage?
Okay. So the first advantage, yeah. So the first advantage is that of time. Oh, but it's an online question. Oh, right.
Yeah. Okay. Yeah, it's an online question.
Both harass karta hai yaar. I mean, I'm not able to even focus right now. Okay. Anyway, so yeah. So the answer to that is, as I said, it takes 15 minutes to half a day to get this answer on AlphaFold. Number one, right? Number two, the models have been so accurate, as I just mentioned, that even the ones that are not amenable to X-ray crystallography or other experimental methods can be solved using this particular thing. So the interesting thing, as I will show you in the algorithm, is that in the algorithm, you can actually build in the biological context. Okay. So if I'm solving a structure by X-ray crystallography or NMR, I have to prepare the sample. When I prepare the sample, how close is my sample to the biological context? The thing is happening in the cell with flares, for example, right? How close is that going to be? So this has always been a question. And AlphaFold actually gives you the ability to program in also the biological context. So I think these factors and the accuracies that they've managed to get at this point are the factors that are the advantages. And the time saving. Equipment, the cost of the equipment, I mean, they were able to do this on a single GPU processor, basically. This right now, that GPU processor costs about 10 lakhs or so, right? But that's still significantly different from lower as compared to the experimental cost. And in the future, of course, these GPU costs will go further down anyways. Yes.
So what exactly did AlphaFold do different that it's able to be way more accurate than all others? What is the difference in this model?
Yeah. So that's what we're coming to now. How does AlphaFold work? Right. This is what we want to do. So these are my last 2 slides where I'll actually be talking about this. It may take us a little bit of time to get through this. So I think, Arnab, because this is a tantalizing question, we can probably have a chai break now. Right. The video, maybe we can show it right after the chai because it's part of the working of AlphaFold.
Okay, so then we're going to finish right now.
I mean, we can take a chai break and then come back. That's what I was saying.
So this is mainly for the online audience. We are going to be, it's been an hour, 20 minutes right now. So we're going to pause the thing right now, the online talk. Of course, those who are in the audience here will get chai and we can come back. If you have questions, feel free to put it on the chat. We will take a look at it even after the session is over. Or you can always send your questions over email to outreach, outreach at tifr.res.in and we will direct them appropriately. So I hope you enjoyed it. Just a reminder, our next session 15 days from now is going to be on ammonia, chemistries and chem histories. And keep tuned to Chai and Y. Follow us on Twitter or Facebook at Chai and Y, and you can always find out what the next session is. Thanks a lot and thanks for watching. See you later. That's only for the online audience. Okay? So yeah, if the chai is there, we can go and have chai. I will then